Hello, everyone, and thank you for joining today's virtual demonstration. Jennifer Marks will be presenting or demonstrating the VS200 slide scanner, and Jen will now take it away.
Well, hi, everyone. I am going to turn this around. I'm working with both Aaron Powery and I believe Alec DeGran will be coming on, if he's not on already, to help us talk a little bit about the VS200 slide scanner. This is a system. It's been residing here in my house in a little corner, so it doesn't take up a lot of space. Its estimate would be about 2.5 by maybe 3 feet for the size of the scanner itself. Without having any interaction, it's a little bit new to me, but we're going to start with going with scanning. So I'm going to share my desktop on Zoom and introduce you to more than just the outside of the system. Okay, so normally when you log in to our VS200 system, you get a screen that looks like this. So this is an exceptionally easy to use system. So we set it up so that we have these very large buttons organized in a manner that should coincide to how you want to use the system. We have single slide options to the left and batch options to the right. To insert the tray, you would simply choose to exchange trays. If there was a loader, you would click the same button, but then you would open the loader door. A single tray system or a 6-slide system opens the door for you and you insert the slides in yourself. When you're done, you tell it, I've inserted my slides. Our slide holders all have an RFID chip in it, so we know what type of slides have been inserted into the system. And to just give you an idea of how quick and easy the VS200 can be, I'm just going to take you through our quick settings. These are settings that are pre-configured to run easily with just choosing your scan projects, and I'll get back to that in a second. You then have to choose where you want to save your data to, and I have mine going to a neuroscience folder. And then with pre-configured settings, you just tell it you want to scan. That's as quick as it is to go ahead and start scanning an image. This particular setting and slide has a barcode associated with it. Now, I personally have come to adore barcodes because it means that the system, I don't have to do any more than load the slides in. It will read the barcode, use the barcode for the naming of the files, and be able to do this all in an automated fashion rather than me having to type in all the information. Or if you have everything in an Excel document, you can upload a CSV file in and populate it that way. But if you do have a barcode reader, everything reads in seamlessly. So this setting, when you do quick, it's going to automatically scan an overview of your sample. It's going to automatically detect where your tissue is located, put a region of interest on it. And then as you saw, those little green dots came up. That's where it placed its focus map positions onto the sample so that it can go ahead and know where your tissue is, where it will focus, and create a topographical map of how your slide or how your section on the slide moved up and down. So we can manage to keep the entire section in focus as you go across it. Now, this setting takes, I think, maybe about a minute to scan pretty much the entire slide in bright field. So we've already done 2 out of the 4 sections, and we're moving on to section 3. So I prefer to look at people as I talk. So,
here for you.
Hello. So as this is scanning, this is a basic bright field sample. If I wanted to, I can go into the viewing mode. I can zoom in and look at samples that have already been acquired. We have a slight delay due to being on the Zoom meeting, I believe. So as it scans and once it's finished, We can come back to the scan project and let it save out. And Rebecca has raised her hand, so I'm not sure what she is asking.
It is all good. Never
Oh, okay,
mind.
you didn't mean
No.
to raise your hand.
Correct.
Okay.
Sorry about that.
So as you're zooming in and out of your image, you can be looking at the data as it scans. Trying to think if there are other things we can talk about. So this is Brightfield. Once we're done with Brightfield, I'll show you how to set up these scan settings, the parameters that we use to automatically detect the tissue, and some of the parameters that we can use for determining where a not so easy sample might be located. Okay.
So basically everything Jen just did is 3 clicks to get an image. And that's it. So if you have relatively standard pathology stains, you just can use quick setting and 3 clicks, and then you just leave, and it pretty much will do that all by itself.
Yep.
So it's very, very fast. It's very easy, and your input is minimal.
So when I said you choose your scan settings, if you'll notice here, we have a bunch of scan settings. They are divided into 2 groups. One is the My Scan projects. These are things that are personal user login specific. So if you have something that you want to use that you don't want anybody else to play with or jiggle with or even know what I'm doing, you can save them to your own user login. The public scan projects are things that you can save out and then have that anybody who logs into the computer or the system can have access to. So these would be settings if you're a postdoc in the lab and you're setting up some settings so an undergraduate can come in later and scan a bunch of slides for you. You put it into a public scan project, it's set up, it's ready to go. All they need to do is load the slides and say go. So that's the difference between My Scan projects and public scan projects. So you can select on one. So if I select on one, you get a description on the left-hand side as to what these scan projects are. These are things that you write in when you save out a scan project. So you put as much information as you need to know later what the scan project is going to do. You have the name, and then you have a detailed description. And this field will tell you exactly what happens during the project. It also shows you how the project's going to be named, where it's going to save the data to, what type of overview you're going to use, and the magnification, whether you're doing a Z-stack or not, is listed in here, and what type of Z-stack will be in here. So you just click on these to determine which one might be the one that is appropriate for what you're going to be doing. For starting out or starting from scratch, I'll just start with our default. So when you do this, you come into the scan settings here, and you can choose first our overview area. So these are a bunch of pre-configured overview and label areas that you can make yourself, but you can start off with either the full slide pocket, which means I have no label, just scan everything. We have some standard bright field settings, a narrow label setting. You just choose the one that you think is appropriate for you. And in this case, I'm just going to come in here, choose your slide, whichever one you happen to want to scan. And again, naming and saving. So where do you want the data to go to? You just select it from your location, and you directed it to where this data is going to go to at a later date. In this case, this slide does not have a barcode, so I have to give it a name. So this just happens to be in position 2. I'll call it slide 2. So one other property that you decide at this point is whether or not you want to do a new image for each scan, or whether or not you want to embed the scan into the overview. And we'll talk about that in a second. So once you do that, you tell it to scan the overview. In the advanced settings, you have the ability to play with your settings. So you can play with your tissue detection algorithms. You can play with the size of the region you want to do, whether or not you're going to be scanning as a whole block, like I scanned that last setup, or whether you want to scan each section as its own region. So these parameters are available to you in expert mode. And once this is done scanning the overview, we can come back in and play with the scan areas. So on the top of our software, you will notice we start from the left, go to the right, scan settings, image, this contrast enhancements capabilities, scan areas are where we play with the scan areas, and focus map, focus map. So you generally start on the left and head to the right. So with the scan areas, I can come in here, and if I choose one for each, the slide, then I get one large ROI with all the space between the regions being scanned. I can also choose one for each subsample. And when I do that, I will get one ROI around each slice. If you see that they're numbered a little bit oddly, we can choose to number them by rows or by columns, and it'll automatically number it for you. Some of the parameters that you have the capabilities to adjust, again, if you want to do some things that are automatically going into another software package where you want everything to be exactly the same size for later, you can go ahead and have everything use the same sized region. In this case, I chose the largest region. I don't want to do that, so I'm going to bring it down. So you can change the minimum size of an object. So if you want to get rid of some of the smaller ones, you can get rid of them. Oh, I got rid of everything by changing the minimum ROI and eliminating one or two or three of these. I want to bring back one. So with sample detection, so this is how well we detect what your sample is and whether or not we consider it to be stained or unstained. So we have a non-colored sample detection weight. We have sample sensitivity here. And if you notice, these boxes will get smaller and smaller as I remove the ability to see this specimen. So I can also remove objects and fill holes in this region as well when you have spaces. So this is how we detect where our objects are in the data. We have some advanced algorithms and the ability to remove markers and things like that. So if you happen to get slides from somebody where they'll look at it on a stereo or on another microscope, and they might circle the section they want you to scan at high mag, you can go ahead and use that marker to find your sample, but also remove the marker later so it doesn't show up in your image. Focus maps. When you have your focus maps, they're automatically put into your sample based on a normal density. Not sure how they come up with that algorithm, but it works really well, just leaving it at normal. You can use a lower density, or you can go all the way up to extra high density for your focus position. So that determines how many positions we need to go to while making that topographical map moving up and down. We generally leave it at normal. In expert mode, you have the ability to edit some of these. So if you happen to be going over a fold or a place where the tissue may have disconnected, you might want to move them in. Or you might say to yourself, this region right here is really important to me, so I'm going to add focus position here. So they are completely editable, movable. You can add them or subtract them. You can even delete all the shapes and start and put them in yourself a couple places if you want. So these are completely customizable for you. And I'm just going to go back and re-add them back in. So this gives you quite a bit of power for being able to scan your slides very well without having to deal with some of the problems that happen when especially novices or difficult types of samples are put down, so that you have a bunch of folds or everything goes up and down. The other option you have is non-sample regions. So if you have something that isn't stained as deeply as this one is, or your sample may not be present in the first 2 slices, but will be present later, we do have the ability to include these areas that aren't automatically detected in our focusing later. That's what some of these buttons are. In our single slide mode, you have the ability to go into either semi-automatic focus detection or completely manual. This becomes more important with fluorescence specimens, but when we go ahead and do batch mode, everything is done in an automated fashion. But in our expert mode and single slide mode, you have the ability to edit these parameters as you go. This becomes helpful with setting up some of these scan properties so you can determine where you might need to place the focus map or parameters that might need to be changed slightly in order to be able to do a good focus map. I'm going to come back to settings. So when you have your detailed magnification, you choose what mag you would like to scan your images at. Our standard system comes with the 2 and 20 for bright field. When you add fluorescence, you get the 4x. And then every other objective that you put on the system up to 6 is user choice. Most people will add a 20 and a 40x air. We do have the ability to add oil lenses to the system, and we have the automatic oiler already built in. One other parameter that you would choose is whether you want to do normal, which is a single Z plane, or whether you want to do virtual Z, and virtual Z is a Z stack of individual images that you can then scroll up and down in. As Erin mentioned in her talk earlier, these slide scans start bringing out quite a bit of data. So if you're having issues where you don't need a Z stack, but you want to see everything flattened on top of itself, then what you can do is get only the EFI out. And that helps you with data management and gives you the final product that you want without having to store and then post-process. So you can choose your range as to approximately how thick your specimen is. Generally, we leave this fairly manual. I don't like to choose top and bottom or my depth of field. I generally let the system make the choices for themselves. I've been doing or working with microscopes for a very long time now, and I think this actually hit my ego a little bit that the machine was better than I can be. But it really is true. I tried to beat it, and it's rare. So if we go back to scan areas just to come in here and not waste everybody's time, I'm just going to come in here and do like half a brain slice. And come back to the settings, do a normal slice, and then you just click scan the detailed image. At any point in this process where I think I've made the changes that I need for the scan project, I can come up to the top and hit Save Scan Project. I can then give it a name, and I can give it a detailed description, much longer description of what this does, and then it saves all the parameters like the overview and everything else that I've selected along the way. And then you can choose whether it's private or public. Since it's private, it will show up under My Scan Projects, and I hit Save. So that's now ready for anybody to use later and apply those scan settings over and over and over again in a batch manner. If I choose to scan, and the scanning, just as a pause, has been a little bit slower on here, probably due to the connection and the resource use for the remote. But you know, half a brain section in 20 seconds, not bad.
Something that we get very commonly asked is how many individual regions can you put on a single sample? And the answer to that is 50.
It's a little bit of a caveat there. The answer to that is 50 if you want to embed them.
Yes, yes, if you want to.
Okay, so what we're talking about with embedding is, as you can see, I have the overview and I have my region that's showing at the same time. So this is an embedded section. If you want to scan it to its own individual file that does not have the overview with it, is just that individual scan. I have not run into a limit to the number of scan positions you have, but you can only do 50 if you want to embed them. Okay. So that is running with a bright field expert mode. What I wanted to show you, I have some pre-planned slides. So if I switch over to viewing mode, I can come in and show you a couple of parameters. So one here is this barcode slide that we scanned earlier. I chose to go ahead and mark it, and it has 4 of the embedded regions that are associated with it. I am going to open another file that is right here. Okay, so with this one, what I wanted to show you is what an EFI looks like and what a Z-stack looks like. These take a little longer to acquire. So if you had a normal 20X and we come in and we look at this image here, you can see that we can see all the neuronal structures showing up. We can zoom in and out. Our actual magnification is shown up here on the image navigator. So we can zoom all the way up to 20X, which is what this was acquired at. And might want a region that actually seems to have a little bit more information. And you can zoom in and take a look at it. There's a pause in our zooming when you reach the true magnification that it was scanned at. You can over zoom, but you're going to be now in digital zoom. So you can migrate around, you can view the data, you can go in and out. When you add a Z-stack, this is when you come in here to this data where it is a complex data set, and you might want to be able to come up and down in Z. And when you click on that, you will get the ability to scan up and down in the Z-stack. So you can scan up and down by using your mouse, or you can use the Z slices over here on the side. They're also present in the viewer up top here. So you can manage to go up and down in Z. The other option that you have is you can do this EFI or extended focal imaging projection, where we project all of them on top of each other, so you see all of the neural tracks all at the same time running in their lines. So this is an output where the output is a single plane for taking the same images that we took in the Z-stack in the plane in the section next to it. So I hope that makes clear the difference between the different outputs that you have. As you can see here in the settings, we have all of the different regions that were acquired on the slide are shown here with eyeballs. And just like, I've got to find some of them that we can turn on and off. So this one right here is the overview underneath it, and then the actual scanned image up above. So you can turn them on and off and view them relative to each other. So this is bright field scanning, and a lot of the parameters I just showed you with bright field scanning are the same parameters that you will have with fluorescence, except we add the complication of determining what your exposure times and a couple of other parameters associated with it. So if you come under fluorescence expert, you have, again, your scan projects, and they're predefined. You can come in here and choose your section that you want to do. You choose your slide just like before. And we can come in here and I'm just going to change it to standard fluorescence. You can decide when you do this overview scan in fluorescence, a couple of different parameters. So one is, how do I want to scan this? Do I want to scan this? Am I concerned about photo bleaching my specimen? If I am, there are a couple of other modalities that you can use to scan. You can scan it in bright field. You can do what people generally consider standard, I have found, which is doing like a DAPI overview of your fluorescence side, or you can use dark field. So just to show you what the scanner does when we do these, I have a couple of these modalities. So here's an overview of a brain section that was done with dark field. You can then add back in your fluorescence overview overlay on top of this. But this is what we used for automatically detecting our tissue, automatically finding our tissue in the overview. So this would be an example of dark field. This would be an example of DAPI. And I managed to go the wrong bright field, and this would be an example of dark bright field. Same slide. So if you have an older section, you may be able to get away with bright field. For frozen sections, bright field may not be the best solution. We may not be able to automatically detect that tissue. And so in those cases, dark field become a very good option if you don't want to use the DAPI and don't want to risk photo bleaching or doing something to your sample in fluorescence. So if we come back to the scan project, we have to make that choice. Do we want to do bright field? Do we want to do fluorescence? If we want to do fluorescence, we have to tell it what channel we want to do the fluorescence on. And you have the option of any of the channels that we can scan on the system. You'll choose what type of sample you have, whether it's dense tissue, porous tissue, or individual cell cultures. And this determines what algorithm gets used for automatically detecting your tissue. Or you can choose dark field. We do have the ability to do dark field both in fluorescence or fluorescence mode, which would mean we use our monochrome camera for it, or we can use our color camera for it. Generally, unless you need the color, we generally keep it with the monochrome. So just for ease, I'll do bright field. Generally, bright field overviews are done with 2x. Fluorescence, a lot of time we switch to a 4x for the overview just so that we have enough light coming in to be able to do that focus map without having to do really long exposure times. We also have the ability to go to 10x, and dark field can be done at 10 or at 4x. So the choice becomes what you would like to use. So again, with bright field, we generally go at 2x, just like we do with our bright field samples. I, again, have to come and decide my name and make sure that it is going to the right location. Okay. And we just scan an overview. Do we have any questions with what we've shown so far? I know when we were, the questions from the previous talk of the day was asking how the system was configured. So this particular system is configured with the Orca Fusion camera. And it has the Xylus light source for white light, and we are using cubes to filter for the fluorescence. So here are some sections of what was automatically detected. As an older slide, sometimes we do have areas that are detected just because they're there. So we may want to come in and change our automatic sample detection and see if we get rid of some of these parameters. And then our scan area will get rid of the rest. Or we can just ROI it. So because this is going to be a scan where we don't need to sit through 7 minutes of just randomly hearing me babble, I'll do a little bit smaller ROI. So when we do the sectioning, again, same choices. What magnification do you want to scan at? Do you want to do a single slice? Do you want to do Z? And now is here where things change. So with the channels, you have the option to add channels or to remove channels. So if you didn't have 2 FITSI channels here, you might want to go and remove one of these channels. I'm going to actually remove both because I have a channel set up automatically with shading corrections for using brain slices. So brain slices are notorious for being hard to get, especially the FITSI to be flat and even. As you receive samples that might be thicker or thinner, some of the shading comes out from the sample or the autofluorescence of the specimen themselves. So I find we can make as many channels as you want, and we associate shading corrections with each channel. So sometimes I find it nice to have a setting that's specific for the type of tissue that I'm going to be working with. When you have these channels, because we have more than one has to be designated as our focal channel, and it also has to be designated as the channel that gets used for stitching. So that is always channel 1. And in most cases, the DAPI works out very well for being the channel that we use for focusing and mapping. We also have the option to do this focus mapping on a different channel that we only use for focus mapping and we don't image. So if I say first channel for focusing only, it means I have DAPI in the specimen. I'm going to go ahead and use it for my focus map, but I don't care about DAPI, so there's no reason to take it and no reason to have it as extra data that I have to store later. So if that's the case, I can just say use that first channel for focusing only. If I wanted to add a transmitted channel, I just click add the transmitted channel and I choose whether I want to use bright field or dark field to add in this transmitted light option. To determine the exposure times, we have the option to just let it be determined automatically. A lot of times with fluorescence, I find it easier to just go around. I know where my specimen is really bright and I will go live and come into the specimen. Oops, my zoom is on top of the specimen. So come in here, you can do a single autofocus and you can do a one-time auto exposure, and that'll come up with a good exposure time that will work for your sample. If I click to the other channel, you can just do a one-time auto exposure. Generally, this auto exposure is set to be a lot brighter than what it needed to be. Is set to be at 2/3 the dynamic range of our camera chip. So although that is a preference that can be changed. And you just run through our different specimens. If you think that there's another area that might be a little bit brighter than where you are, you can click and move to that area, or the system comes with a joystick that I'm moving right now using a joystick, so that you can use wherever you think your brightest area in the specimen is to determine your exposure time. So once you've determined those, you can just stop live. And I'm going to just reduce this down a little bit and scan. So while this is going ahead and doing its mapping and scanning, the number of channels that we do, we're seeing a lot more of the size 7s and a lot more requests to go further out into the IR. Generally, so far, we've been able to meet most of those requests, and 5-channel, 6-channel imaging is not unusual using the system. So as it continues through its scanning, you can then go home. And I did want to show you a couple of options again with some slides for fluorescence, for the different options for how this scans might take place. So the first thing is, here's a Z20X. They're just some sample slices that we were given. If you wanted to work with this Z, you can scan up and down it. Interactivity is a little bit to be desired, but you can scan up and down into, and let me stress that's due to Zoom. So as you scroll up and down in the slice, you can see it come in and out of focus. So this would be a Z stack of just wide field. One other option that we have is we can set up the system to have what's called online deblur, which is a 2D decon that gets applied as we are going to acquire the sample. So you can get an image that will look something like this, and you can zoom in and you can see very detailed settings that we can come back to later with this Z stack. So you can then scroll up and down, and now when you're going up and down, it is our deconvolved data. So you can see some of, okay, it's updating rather slowly. So you can see some areas that are in focus above and below, and that we can put them all together and view them as we need to. That is an example of using the online deblur. We also have the option of doing a maximum intensity projection. So when I use the deblur, I find that this is one of the prettiest ways to take a look at the data. We flatten it all into one image, and it is all here and available, but it's a single plane. Again, data management is really why you do that. If you don't need the Z, you don't need to acquire it. If you want to use the Z and do post-acquisition deconvolution, which is a package we offer as well, you can acquire those Z stacks and then deconvolve them offline. Both are options, it's just a personal preference. This is an example of maximum, before I showed you maximum intensity projection, this is an example of extended focal imaging. With extended focal imaging, what it does is applies a focus map, and it considers what's in front of the, in the stack as to what you're viewing, and projects them together to give you your three-dimensional view of a section. So these are options for how you can use the system to scan, and those were just check boxes on the scan. Do I want to do a virtual Z, a maximum intensity projection, or an extended focal image? We had mentioned before that we had a lot of different slide tray options. So we have the 1x3, which is what I have been using. And the system, I'm going to exchange the trays. I'm just pulling out my 1x3. Here's my 2x3 slide. And when I put that in, the system will automatically know what I've loaded. Okay, so if I go to Fluorescent Expert here and go into my overview, it now knows that I have 2x3 slides loaded. If I select this image, one of the things that we've been talking about quite a bit is the ability to do multiplexing. So multiplexing is more than one channel, but really it means more than just doing 5-channel imaging. It means I've done these 5 channels, I've taken my slide off, I've then inactivated those channels, restained for new fluorophores, recover slipped it, thrown it back into the machine again. And now I want to be able to image these same sections again, looking for new dyes, new, maybe the same dyes, but looking for new proteins and where are they localized at the same time. So now I'm not looking at 5, now I'll be looking at 9 different things at the same time. And you can do this over and over again. The way we make this easy to do is you can reload and reuse overviews. So you just reload, reuse this overview, it populates the same slide that was there before. So now if I come in here and I choose to scan the overview, I have to tell it I want to go to my actual folder. It just has the overview you previously did. It has your previous sections. And now when I want to do my detailed magnification and scanning, I can use these same parameters or slightly different ones because I didn't save them, and rescan the same regions. Right? So if I come back in here under scan areas, this was the last one I scanned, and I can just draw as many regions as I want and rescan those same regions a second time, looking for new options. So when we do these sections, I just wanted to show you real quickly how you would do the deblur. So if I turn this on and we look on top of my section, and here I am. I don't even know if I'm on anything. Let's go to depth. Okay, if I just click this button, deblur, that's as quick and easy as it is to do the online deblur. So you have to activate it for each channel, but then it becomes activated overall. I'm going to stop live And I'm going to exit out of this right now because one thing that I do think, we've been talking about single slides, but this actually is a 6-slide system. And we have a loader that goes with it. The loader actually would look like this, except another unit right next to it at about the same size. And that brings us to the batch over here. So once we've set up these settings, usually done with a single slide to determine which settings work best for my samples, you then want to apply these settings over and over again to all the different slides that you come up to. So you would just come into your expert batch. You would then choose your sections you want to apply, whichever scan project you're using. You'd say edit the overview. And then I can define where my batch is. And in this case, it's doing 3 slides, but I happen to know that's transmitted. So you can confirm that this is what I'm going to apply in my batch. So this is a 2x3 slide, but if I had the loader next to it, there are 35 different positions in the loader. So you would get 35 options here. And then when you click on the different positions for the trays, you would then get a preview that looks almost identical to this. So you would get the trays and it would know exactly where your slides are loaded. And then I'm just using a scan project that I previously used. If I wanted to then, let's say I had another tray, I can come in here and load a different scan project to be applied to a different tray. So you would have this one and you would have that one, both being separate scan projects. And so in that way, if you have 2 people, you may not have 210 slides with your project, but one person may have 40 slides and another person has another 30 slides. They can both apply and load them in and have one overnight scans session where they each are able to use their own slides and their own scan projects. That makes sense? I'm not sure if we have any questions or not.
Not yet.
There's one other thing that I think is important with the workflow, and just because I have 3 slides over here, load those back in. One other thing that is very nice when you're doing the batch is that you can start working on your scan projects and your overviews while it's still scanning the next overviews. So if I define my batch project now as the middle 3, I can confirm them. I'm going to still use the bright field. And I'm going to go tell it to go ahead and start those. Start saving to the correct folder. And then no. Now I can go ahead and start scanning my overviews, but I can start editing my overviews and my regions of interest and my focal positions on the first slide as soon as it's done scanning that slide. So as it's scanning the next overview, I can go ahead and edit the existing overview and continue doing this so that by the time I'm done doing my 30, 40, 210 slides, I've gone ahead and edited and made sure everything is perfect on the overviews before I tell it to scan. So we don't have that delay in the scan project. So I can come in here, it's scanning the next overview, and I can come in here and play around with the views and decide where my regions of interest are. Go ahead and switch on my focus map. See where it's mapping and saying, oh, that's not on my tissue. We're going to delete that one. I want to move this one up. Okay, I'm ready. I'm going to the next slide. And then here's this overview. Oops, previous slide, finished it already. This one it auto-selected perfectly and put some really good focus positions on. So I'm pretty happy with it. Now I'm going to go to the next slide, which it's scanning. So your workflow when you start doing batches becomes very streamlined. You've defined your parameters. You've set up a scan project. You then load all your slides in, define which project you're going to apply to each slide, tell it to start scanning the overviews, and then start editing your overviews. By the time you're done, you can just go ahead and tell it to scan all your detailed images out. Do we have any more questions for this? I kind of left it so there would be some room for people to ask questions, or maybe see where we can go for now, because we've reached about 45 minutes. So do we have anything else?
Jen, we have not received any questions.
Okay.
I think that was a really comprehensive review of the basic system functionalities. And we're using very standard samples here, but I know that Jen and I have both seen some really unusual things being scanned on this. So it's always very fun for us to see what you guys actually bring to the table. I've imaged through a 300 micron cornea with this machine, and I was absolutely amazed at how well it managed that incredibly challenging sample. I know Jen has seen some really interesting things. She's imaged crystals, like household crystals before, in polarized light, and they're incredibly beautiful. So continually impressed by how flexible this system really is.
I think the only specimen I have not been able to image that somebody has sent me was a bright field sample that came on cardboard, so I couldn't get transmitted light through it. Other than that, I've been able to pretty much scan everything I've been given. Some of them have required putting on a long working distance 20x phase lens. We have done a wide range of images, samples. It's been interesting for me, honestly, to see how much polarized light can have influence on different types of samples, especially, in clinical research. People are discovering that viruses or some of the bacteria can be seen in polarized light, which may make it easier to find them and scan them at higher mag. So there's been a lot of interesting things that we've come across. Brain slices most often get scanned at, and one thing we mentioned with reloading the overviews and being able to scan again, a lot of times people are doing things looking for clones where they've either injected them or they're somehow got either cells or something into the brain of the mouse, and they're looking to see where they are localized, and they don't necessarily know where they are localized or where they might be co-localized. So they'll go ahead and do scans at 10x and then come in and look at higher magnification, whether that higher magnification means a 60x oil or maybe a 40x air. They can then zoom in on where they are for a higher mag study. So I see that an awful lot with brain slices, just where in the brain, let me map where these things are located, and then trying to reconstruct that later and be informative. So a lot of these data, we didn't talk on any of the analysis other than mentioning that we have deconvolution. A lot of this data gets taken out into packages that will specifically analyze and be able to look in detail, whether that is Neuralucida or taking it into Imaris or Visiopharm or Halo or any of these programs that can deal with whole slide images. It's very easy to do from these formats.
All right, any other questions?
Doesn't look that way. I would like to thank both Jen and Erin who are both research imaging specialists with Olympus, for showing us the VS200 and speaking to its applications. This session will be available on demand starting tomorrow on the Neuroscience Week page. And Neuroscience Week has only just begun. Be sure to check out www.neuroscienceweek2020.com for tomorrow and Thursday's lineups and to enter our coloring contest. The next event for today is a special session on single cell manipulations with a nanosyringe presented by Cytosurge. Registration is still open through the Neuroscience Week website. If you have any questions that may come up after you've left this demonstration, you can post to your social media with the hashtag Neuroscience Week 2020, and we will answer them on the social platform. Thank you for your time.
Thank you. Bye, everyone.